presenting on this paper titled Application of an Integrated Computational Antibody Engineering Platform to Design SARS-CoV-2 Neutralizers. So in this study, the authors applied computational antibody engineering techniques to 3 different antibodies known to bind the SARS-CoV-1 receptor binding domain. And their goal is to make them bind the SARS-CoV-2 receptor binding domain. And they came across these 3 antibodies because they had experimentally validated binding to the SARS-CoV-1 RBD. And they reasoned that a combination treatment of engineered variants of these antibodies could be efficacious in therapeutic or prophylaxis against SARS-CoV-2. The idea being that multiple antibodies on the same targets will decrease the chance of mutational escape. So they established a computational workflow, shown in Figure 2, to increase both the developability and the binding affinity of these antibodies to the SARS-CoV-2 RBD. Initially, they obtained the FV regions of all the candidates by extracting them out of the PDB complexes where they originated. And they looked at developability features like surface patches, chemical degradation of aspartic acid and asparagine, and methionine oxidizability. They used Discovery Studio and Mo to do this. Prior to any of their computational work, they protonated and energetically minimized their structures within Mo and Rosetta. And to begin their affinity maturation protocol, they obtained the standard PDB complexes of these antibodies bound to the SARS-CoV-1 RBD, and they manually replaced the SARS-CoV-1 RBD with the SARS-CoV-2 RDB in Pymol, and then relaxed the structures and began the computational workflow. And they used 4 tools to generate interface mutations. Mo, Rosetta Flex DDG, TopNet tree, and SAMBI 3D. The latter 2 were machine learning methods. And so in all of these methods, all antibody residues within 6 angstroms of the RBD were mutated with all 20 canonical amino acids, and DDGs were tabulated for all of those positions, for all those point mutations. A DDG z-score was then averaged across the 4 methods, and the top 60 average scores were used to select the final list of affinity-enhancing mutations for each of these 3 antibodies. For library design, they didn't do any expression, but they estimated that the theoretical library size would be suitable for phage display, so 10 to the 11th was their upper limit. And interestingly, they mentioned that they included NNAS glycosylation on the FC region to minimize antibody-dependent enhancement, and also DQ mutations to improve antibody half-life, also on the FC region. Thank you.